I'm going to wait 1 second because I'm hoping they come with the card.
So I was going over everything. And I was thinking, like, all right, so memorize the trees, I picked up a billion characters, and then I hit the question that's like, I'm a study guide. You're not expected to know everything. Bacteria,
yeah, all right, that was a really hard one. I should not have used that.
So here's what I was thinking.
That's too.
I was just going to make a suggestive, and the question you know. But what I thought was really good was on the practice system. Instead of saying, we're checking to see if we do one little minute code check, you ask us to give examples.
Yeah, no, right.
That was cool. I thought that was
Yeah,
because then
so it's a balance, because those are hard to grade.
right, yeah. They're hard to grade, but like it's easy.
Yeah. The test is already written, so.
Okay. No point out. Right.
All right. Gonna get going. Reminder, I had sent around an email about this, but I didn't send around another one. We're going to have another review session tonight from 4:10 to 5 in 2 Wellman. And then tomorrow, I can't remember what room it's going to be in, but it's going to be from 5:10 to 6. Yeah.
You're going to post the answers to this practice test?
Answers? Yeah, I was just letting people sort of think about them. I will post answers later today. So. But I wanted to sort of force you to talk about them with each other and not just sort of go through and think you know the answer. Because whenever I used to get answers for tests, I'd be like, oh, yeah, that makes sense, without actually thinking about it. So again, for the review session, come there with some specific questions about something you don't understand. I'm not just going to review whole lectures. If you have a question of something you don't quite get, then I will answer those questions. So what we're going to do today is talk, as I said before, about, can you stop talking? All right, thank you. We're going to talk about extremophiles and also about sort of how you study microbes. And then in the next lecture, Friday, first lecture, we're going to talk about human-associated microbes, building on many of the methods that I talk about today for how you study microbes. But the types of organisms we're going to focus on today are more the extremophiles. And I just wanted to point out something which I didn't completely sort of link together, but one of the themes for many of the lectures and many of the topics that we have talked about has been this issue of creating chimeric organisms. So lateral gene transfer can move DNA from one branch in the evolutionary tree to another branch. The recipient of that DNA now has different parts of its genome having different evolutionary histories. That's called, sometimes referred to as a chimera. It's named after the sort of combined organism from Greek mythology. Recombination, sexual recombination in eukaryotes is also, in essence, something like this. DNA gets mixed from different lineages. In most cases, that happens within a species. In fact, that's sometimes how we define what a species is. You can have sexual reproduction between closely related species, hybridization events. But in general, you don't have DNA moving across large distances, but over short distances, it creates the same effect. You have, say, two different populations that come back together, and the DNA mixes from those different lineages.
Question?
Yeah?
Are some of the unicellular eukaryotes capable of the lateral gene transfer mechanisms of bacteria and archaea?
Yeah, this is a great question. So we don't know a lot about all of these unicellular eukaryotes because many of them, the genetic processes that they undergo out in the wild are not very well understood. But yes, they in fact do undergo lateral gene transfer, much like bacteria and archaea do. It appears to be a little less frequent in many of them than it is in bacteria and archaea, and maybe, although not entirely clear, the DNA might be moving over shorter evolutionary distances most of the time. So eukaryotes, in some ways, are not very good recipients of foreign DNA from bacteria and archaea because they have introns, they have a nucleus where the transcription occurs, and then the RNA has to get out of the nucleus to be translated, and some of the signaling for that is pretty different than the signaling for what goes on in bacteria and archaea. So a piece of DNA from one organism inserted into another doesn't always work in that other organism. They have to, transcribe it and translate it and make it into a product. So movement of DNA between close relatives works much better than movement of DNA between distant relatives. It turns out that lateral gene transfer can happen in anything. It just seems to be more common in bacteria and archaea. And one really important example of this relates to this other mode of creating chimeras, which is endosymbiosis, when a eukaryotic cell brings inside of it bacteria, for example, like in the history of the mitochondrion or in the history of the chloroplast. What has happened over evolutionary time is after that sort of relationship gets stabilized, and then you have an organism that in essence has two genomes, a nucleus and a mitochondrial genome. A lot of the DNA that's in the mitochondrial genome over long evolutionary timescales moves into the nucleus. And the genes that are coded for actually get made in the nucleus and frequently transported back to the mitochondrion. Same thing happens in chloroplast evolution. Because they have to acquire sort of eukaryotic signals, you have to, again, transcribe and translate in the way eukaryotes transcribe and translate. I'll get to your question in a second. That process appears to be pretty rare or slow for the DNA that, say, makes a part of the mitochondrial membrane. The gene for that may eventually move to the nucleus, but it's rare because it has to sort of acquire all of the processes for transporting the encoded protein back into the mitochondrion. But it does happen, yeah.
When you look at genes which stay in the mitochondria and genes which leave the mitochondria for the nucleus, what sorts of genes are the ones that are staying and which sorts are the ones that are moving?
For the mitochondrion, which is a symbiosis that happened basically billions of years ago, there are very few genes left. There are a couple of lineages that have been studied where the mitochondrial genomes have maybe 100 genes. Most, like animal mitochondrial genomes, have about 15 to 20 genes usually. And the things that are left, it's somewhat sporadic. There's not always the same in different lineages. The things that are left appear to be, there's a theory about this that suggests that they are the the genes that for some reason can't get transported from the nucleus
Divide the mitochondria.
into the mitochondrion. So they might be making proteins that are really hydrophobic, for example, or they need to interact with some other thing while they're being made. And so that the theory is that there may just be a small set of things that have to stay in the mitochondrial genome.
And
Yeah.
the replication genes stay inside the mitochondria.
No, actually, many of the genes involved in copying the genome, the DNA polymerases and things, they're actually in the nucleus. So that gives, in essence, that you can think of this as that giving the nucleus sort of control over the replication of the system. Yeah.
So mitochondria, let's say there used to be bacteria with, say, 1,000 genes.
Yeah.
Did most of those genes get into the nucleus, or did most of those genes just disappear from somewhere?
Yeah, so with everything in evolution, events that happened a really long time ago are very hard to figure out exactly what's going on. If you ask the same question about the chloroplast instead, which is a somewhat more recent evolutionary event, it happened just in the lineage leading up to the Plantae group. So it's, 1 billion years old instead of 2 billion years old or something to that effect. You can actually look for much more carefully for what genes are left in chloroplasts and what have moved to the nuclear genomes of plants. I actually did the first analysis of this because I helped sequence the first plant genome. And when we looked in this model plant genome, Arabidopsis thaliana, there are thousands of genes in that genome that came from the chloroplast. So it appears that a huge fraction of the cyanobacterial genome is now inside the plant nuclear genome, making things that probably are involved in something to do with chloroplast function. The mitochondrion, people have tried to figure this out by looking for nuclear genes that are related to alpha proteobacteria. So you look in the nuclear genome for genes that appear to be closely related to alpha proteobacteria. And for a variety of reasons, this turns out to be harder to do than to look for the chloroplast-derived genes. The estimate is maybe about 500 of the genes that were in that proteobacteria are left in nuclear genomes, but the estimate is like 500 plus or minus 4,000. So it's not a very precise estimate. But if you can also look at all the proteins that get sent to the mitochondrion, that is what are transported into the mitochondrion, and there certainly are thousands of things that go into the chloroplast and mitochondrion. Some of those may have been in the nuclear genome, some of them may have moved. It's an area of research now. We're just sort of starting to get a handle on it.
One other question. Is it an advantage to transfer genes to the nucleus because they have sex?
I mentioned before the theory that is trying to explain why some things get left in the mitochondrial genome. You're exactly right. Most people think that over evolutionary time, the genes that make it into the nuclear genome are somehow fitter. They're somehow either more stable. The replication processes of the nuclear genome seem to be a little more accurate than those in the mitochondrial genome. And then you have sexual recombination to mix and match things. So it is thought that that is the selective advantage for things to move to the nuclear genome. But, it's complicated in some cases because sexual reproduction is not always advantageous. There are advantages to clonal reproduction in certain situations. So it's a balance. In some lineages, moving to the nucleus may be good. In others, it may be a bad strategy. Okay, so we're going to shift now to talking about this sort of combined topic, and I'm going to mix and match basically information about studying extremophiles with some information about how we actually study microorganisms in the world around us. So if you go to any environment, I've picked these extremophile environments because that's what we're going to talk about today, but if you go to any environment and you collect a sample, a sample from your scraping of your skin or your teeth, or in a hot spring, like this is a overhead view of one of the hot springs in Yellowstone National Park, or you collect pond water, or wherever you go, you see microbes when you look in the sample with a microscope. You can go to, again, these cool extreme environments, And surprisingly, people sort of didn't expect this in many of these environments, but if you go to those environments, they're chock filled with microorganisms too, including in the bottom of the ocean, in boiling hot springs, in acid pools, and other places. And so you look at them in the microscope, you can characterize the environment that they come from. So you can look at, say, this is, that particular chemistry. This is monolake, some other type of chemistry. This is a picture from a postdoc of mine who was in Antarctica studying some microbes there. And you can have a feeling for what might be going on in those environments by sort of looking for microbes and looking at the environmental conditions. And like I said before, there's incredible diversity if you look in the microscope at the form that you see in microbes. So you can try and do this comparison of what you see in the microscope to what the environmental conditions are. And in general, when people do this type of thing, really oversimplification here, but we can think about the questions that they might be asking in these two categories. Who are they? That is, you go to an environment, there are microbes there. What type of microbe are they? What lineage are they from? Can we use that? We classify organisms to help us understand and interpret when we compare different environments, etc. So one key question is, who are they? And then a second key question is, what are they doing? So if we observe these microbes in a particular environment, we'd like to know what functions they are carrying out in that environment, whether it's in our gut or in a hot spring or whatever. And what I'm going to do today is sort of, at a very big, high overview, talk about some of the approaches to doing this using extremophiles as an example. The general approach can work for any microbe, that is bacteria, archaea, or microbial eukaryotes, and it can work for any environment. So I'm going to talk about just sort of three categories of approaches to doing this. We can go out into the field and do field observations of microorganisms and their functions. We can take microbes from the field and grow them in the laboratory and study them in the lab. And then we're going to talk a little bit about what I call Csi Microbiology, which is basically to use analysis of the DNA of microbes in the environment without growing them in the laboratory to tell us something about what's going on in that environmental sample. So if we think about observations directly in the field, if you have you've taken courses where they've talked about ecology and evolution, and field biologists do this in lots of different ways. You can go and collect butterflies. That's what I used to work on many years ago. That's not me, by the way. That's Vladimir Nabokov, the author, famous author who also happened to be a well-known lepidopterist, going and collecting them in the field. You can, set up a plot in a particular area and characterize the organisms in that area as well as the functions that they might do. We now make use extensively of things like satellite imagery of particular areas to correlate, say, observations of chlorophyll or other parameters to what we see on the ground. We can do structured setups in environmental samples. This is like a quadrat where you divide a sample into a grid and you characterize the things in the different parts of the grid. You can get confused and not know where the bird is, but you can go and do sort of close-up observations, although you can facilitate them with things like binoculars, and you can go to all sorts of different environments to do this. People are very familiar with this for doing this for plants and animals and other macroorganisms. They're probably less familiar with these types of things in microbes, but each of these approaches people have used to study microbes in a variety of environments. So you can go, just like people collect butterflies, you can go and collect microbes. This is a hot spring in Yellowstone National Park, and you don't want to put your hand in there, but you can collect samples from that system and characterize them using some of the same principles that you would if you went out and collected butterflies from an environment. You can sample in the field, set up a particular sampling protocol. This is from a trip that Craig Venter, who also worked on the human genome, sailed around the world collecting ocean water samples and characterizing the microbes in them. We use satellite imagery a lot to study microbes in the environment by looking at things like chlorophyll absorbance or other absorbances of diatoms and their relatives or other microorganisms that photosynthesize. We can set up those same type of quadrats to characterize the microbes. We're going to use a microscope instead of binoculars, but it's the same general idea. And we can do this in all sorts of different environments, like boiling hot volcanoes in the bottom of the ocean. And this type of approach is used extensively in what you could call field microbiology. So if you wanted to do this, and you wanted to figure out who is out there when you do these field observations, if you do this for butterflies, you can observe what they look like and try and classify the butterflies that you see. Occasionally there'll be a new species, but in many cases you can use information that people have accumulated in a field guide or something to that effect to figure out what butterflies or birds or turtles or whatever are in a particular environment. So we can do that with microbes. We can look at them in the microscope when we collect different samples. We can observe their behaviors in the microscope, and we can try and identify them from their appearance. For some microbes, that works okay. But as I've told you already, for many microbes, in particular bacteria and archaea, their physical appearance, like you would see in a microscope, is not a very good indicator of what kind of organism they are. And therefore, this approach is very hard to apply for many microbes. It works for a lot of microbial eukaryotes. Diatoms, we actually have field guides to diatoms. A variety of other microbial eukaryotes that have complicated morphological structures. We can use similar approaches as we use in plants and animals. But for a lot of microbes, such as most bacteria and archaea, this just does not work, or does not work very well. I'll come back to what we do about that in a minute. So we can also do field observations of the functions of the microbes in a particular environment. So you can go to a hot spring, and just like you might go to a forest and measure photosynthesis in the forest, or primary productivity in the forest in some way, or nitrogen metabolism, you can do the same things. You can use satellite imagery. This is a satellite imagery of some glowing bacteria, bioluminescent bacteria. We can use this, for example, to measure nitrogen flow in certain ecosystems, and so on. So we can try and infer functions that are found in particular environments where there are a lot of microbes. We can measure absorbance of particular wavelengths. We can measure how that changes over time. We can measure carbon dioxide and carbon monoxide and sulfur and other things in particular environments and try and connect it to the microbes we observe. We can do this on a fine scale, like a small pond, or a big scale. There's lots of really interesting field work in microbiology. One of the biggest problems with this is that issue of the appearance. So if you do a field observation or a field study of a forest, you can observe what trees are in that forest, or what the ground cover is, or what the squirrels are in that forest, and make some connection to your functional measurement. Again, for microbes, this doesn't always work. So we might be able to measure something about the function, but it might be very hard to connect that to what organisms are in that particular environment. In addition, because they're small, some of the functional studies that you would like to do, say the functional study of an individual tree, studying a single microbe in an environmental sample is tricky. So there are some complications with doing this. And this is just an example of a type of question we might ask about this. I've sort of already explained this, but I guess we can go through it. So which of the following is a reason why morphological traits of bacteria and archaea are not useful for determining what type of microbe one sees in a sample? They're not genetic. They're prone to synapomorphy. There's a lot of homoplasy of morphological traits. Or bacteria and archaea have no morphological traits. Yeah, so C, the issue with microbes is really that the same phenotype, the same character trait, evolves over and over and over again in different lineages. And they change back and forth between different properties very frequently. So in essence, this creates homoplasy and makes that feature not useful for making phylogenetic inferences.
Question?
Yeah.
When you were talking about when you observed what is out there via field observation, I was wondering why it's difficult to
Why it's difficult to what?
Why it's difficult to view them under the microscope or something.
It's not difficult to view them under the microscope. We can view them under the microscope. The problem is that 300 different lineages of bacteria look exactly like this. And 800 different lineages of bacteria look exactly like this. This doesn't tell us enough information. It gives us, whereas if you observe a bird in the field, you can figure out that it's a bird. Occasionally you might make a mistake. I used to work on hummingbirds out in the mountains in Colorado, and one time from a distance we thought we had seen a hummingbird doing something really weird. It was at the wrong flowers, and we got closer and it was a sphinx moth, which has converged in some ways to the flight behavior of hummingbirds. So from a distance we made a mistake, but in most cases you can figure it out in the end what type of organism you're seeing for birds and animals and trees. For microbes, you look in the microscope and it doesn't help for many of them. That's the problem, because first of all, there are too few traits, and the traits just are present in so many diverse lineages, it's not informative. Now again, I've said, and I'll repeat it, this is not true for all microbes. There are some that we are starting to have good sort of what you could call field guides. This again is mostly the microbial eukaryotes that have good morphological features, diversity that we can track with microscope observations. And another problem again is that you just may not have enough material to do some type of functional study like measure photosynthesis in a single cell. So what people do a lot of for the history of microbiology and still today is grow microbes in the laboratory. So we go to a field sample and we take that field sample back to the lab and we try and grow the organisms from that field sample in the laboratory so that we can study them in more detail. And this is generally known as culturing. So culturing is the growth of microorganisms in controlled or defined conditions. That is, you make up some solution to feed them, or you make a plate, like many people use, that you try and get them to grow on. And then there's a very special type of culture that has been very important in microbiological studies, which is called a pure culture. And a pure culture is really the ideal, and it's one where you're growing just a single microbe in the sample. So if you go to an environmental sample like a pond, you can bring it back to the lab and try and grow it in defined conditions. And if you just do that, sort of put your pond sample into defined conditions, you're probably going to get thousands of things growing. What people try and do is separate out individual cells from the environment and get individual cells in isolation to reproduce, and then you have a pure line of that microbe in the lab to study. So what you do is you collect a field sample, you provide specific growth conditions, if you can. So you define what the energy source is going to be, what the electron source is going to be, what the carbon source is going to be. That goes back to all the trophy patterns that we talked about before. You have to add other things in many cases that were not in that sort of trophy list that I made. So various micronutrients like elements, salt concentration, pH, oxygen conditions, etc. And in essence, you dilute the sample out over either many sort of dilutions, or sometimes you can do this all at once, and you try and get a sample where there's just one cell of a microbe in that sample. And then you hopefully get it to grow in the laboratory. Yeah.
So basically you're naturally selecting for that, or artificially selecting for that by providing conditions that enable it to outcompete the other microorganisms?
Yeah, so sometimes you do it that way. Sometimes you provide certain conditions, and then you, in essence, do a selection experiment where some things will grow and survive in those conditions and other things will die. That works very well in many cases. It usually doesn't eliminate everything. You usually end up with a still mixed culture after that. So what you then try and do is you literally try and set up what's called a dilution process, whereby you can end up with single cells in your tubes. And there are a lot of different ways to do this. One, if you've seen it, people do this on plates, these little sort of agar jello plates. And you can take a scoop of your sample, put it on your plate, and then run a little stick along the sample to drag a tiny bit of the sample along your plate. And then you run the stick, a separate stick that's clean, on the sample again and again and again. And eventually, if you do this enough times, there's just a couple of cells on the last little streak that you do, and you can get individual cells. Another way to do it, which we actually do a lot of now, is to literally look in a microscope, and you can use what are called optical tweezers and pull cells into different compartments. Yeah?
How do you know that if you're not using the optical tweezers method that you're getting the specific cell that you're looking for?
You have no idea. This is very hard. And if you go to an environment, what you do frequently is you pick the conditions that you're interested in. You can get all sorts of stuff. So we do this, from soil samples, and you can say, I want to get things that are aerobic, that is, they use oxygen, and they feed on cellulose. And you'll get 8,000 different things when you do this. So you have to do sort of series of this if you really want to get a particular thing. Usually, it's very hard to specifically get one type of thing when you do this. What you do is you get many, and then using the methods I'm going to tell you about in a second, you characterize the different things that you get. Why, thank you. All right, thanks for pointing that out. So this is an example of some of the ways that you do that. This is this plate streaking method where you take a scoop of your sample and streak it out here. So there's billions of cells there. You streak across again, you streak across again, you streak across again, and each time you're diluting the sample. And eventually you end up with single cells being scraped across this plate, and then you get what's called a colony. And these little bumps of growth there, called a colony, are coming from a single cell that was sitting on that plate, and then it grew up into a little pile of cells. The way this works, whether you do it in liquid or on plates, is basically taking advantage of that binary fission process that I told you about. So bacteria, archaea, and many microbial eukaryotes will reproduce asexually, either with bacteria and archaea by binary fission or with eukaryotes by mitosis, and they make copies of themselves, like I talked about before. So you have one cell, makes copies of itself and copies of itself and copies of itself and copies of itself until you have billions, and then you can see them like on that plate as a colony. So you can then take those billions of cells and study them a little more detailed than you could with a single cell. Or you can do this in a test tube, and you can grow up 5 milliliters of some particular microbe. That's trillions of cells from a particular microbe, making copies of itself. Now, you get mutations during the growth of these cultures, but all the cells at the end of that are closely related to each other if you started with a single cell. And that's enough material then to do all sorts of experimental studies with your organisms. So now it allows you to study processes in those samples and connect a process to a single type of microbe, and allows you to do the diversity of things that we do in labs around here, whether it's genetics or physiology or genomics or whatever, because now you have large volumes of, in essence, uniform material to study. You can now look at the microbes more carefully in the microscope. It's probably easier to study billions of them than it is one in a pond water sample, but you still have the same problem, which is the appearance doesn't necessarily tell you what they are. So you can grow all of these in the lab, study their physiology, and you still might not know what it is, which is annoying. So what people do is then they try and classify the organisms that they're studying. And there are many different ways to do this, but the standard approach today is to sequence ribosomal RNA genes from those organisms and place them into the tree of life that we have for ribosomal RNA data, where we have data from lots of other organisms, including microbes that have been studied. And then for your new one, you figure out where it sits relative to ones that have been characterized. So the way this works is you go to your sample, you extract DNA from the sample. You have DNA in your test tube, so there are various processes that we can use to get DNA out of a culture of bacteria or archaea or microbial eukaryote. We somehow enrich in that sample for the pieces of DNA that correspond to the ribosomal RNA genes. When Carl Woese did his original studies, he actually extracted out of cells their ribosomes and looked at the RNA inside their ribosomes. And the reason you want to do this is there are lots of pieces of DNA and RNA inside a cell. And we want to focus in on this particular part, the part that makes the ribosomal RNA. So you want to enrich that in some way and make your sample have lots of ribosomal RNA genes from the organism of your interest. And then, using methods we're not going to talk about, you can read the sequence of letters in that piece of RNA or DNA, the string of letters. That's called sequencing. So here's an example of a string of letters you would get out from one of these experiments. You then compare them to the ribosomal RNAs from other organisms. We make an alignment, that is our data matrix. We talked about this before. You're lining up the different parts of the ribosomal RNA with each other. And now we have a data matrix, and we can build an evolutionary tree. And we can figure out where our organism sits compared to organisms that have been characterized. And that allows us to classify it. Yeah?
So when they were talking about, like, did they look at the 2D and 3D RNA structures? Is that how they characterize? Because I thought you talked about that like in previous sections. They look at the 2 and 3D structures of that RNA to like get that.
Yeah, so imagine you, the way I talk about this usually is by some type of analogy. So imagine you're studying vertebrates, and you go to a place, and there's just a pile of bones there. And you want to compare those bones to other vertebrates. You try and line them up in terms of a skeleton. So you find the humerus bones, and you line it up with other humerus bones, and you use the structure of the organism to help tell you that you're in the right place. So you hopefully don't line up wrist bones with ear bones and things like that. We do the same thing with ribosomal RNA. It's a string of letters, but it folds into this particular shape in the organism. We can predict that shape pretty well, and that allows us to line up different parts of the ribosomal RNA from different organisms, because this little loop in the structure should line up between all organisms. So we do the same thing you would do basically with skeletons, but now with DNA data. And that's what allows us to create this data matrix such that the columns correspond to homologous positions between organisms, just like you want to have the bones lining up in different vertebrates. So that's basically what Carl Woese and everybody else has done for characterizing microorganisms and placing them in a tree of life. This is now required if you want to describe a new organism. It explicitly says you have to sequence the ribosomal RNA genes, build a tree, and say where it sits relative to other organisms. So this is a key part of studying microbes to figure out what they are. And again, in many cases, when you look at them in the microscope and then you build one of these trees, the tree puts something in a place that you would not have guessed from the appearance. The appearance is frequently misleading as to what type of organism you're looking at.
What happens if you can't culture it?
We will get to that in 2 minutes, or 5 minutes. So it's a great question, but I'll come back to that in a second. So what people do today is a little simpler than what Carl Woese did, and this will lead into what you do if you can't grow them in the lab. So you do the same thing. You go to your cultured sample, you extract the DNA, and then there's this special chemical reaction called PCR, or the polymerase chain reaction, which allows us to make specific copies of a particular piece of DNA from an organism. And we make billions of copies of the ribosomal RNA genes from that organism in this one chemical reaction in the test tube. We don't have to extract ribosomes from the organisms anymore. We just run this reaction. It makes the copies for us. And then we do the sequencing, and everything else follows the same protocol. But this greatly simplifies studying these cultures of organisms in the laboratory. And this is what has allowed people to characterize all sorts of different environmental samples like the ones in the human gut. Yeah?
So that's if you don't have access to a laboratory, is what you're saying?
No. You do this with organisms you grow in the laboratory. You extract their DNA, and this reaction allows you to make billions and billions of copies of their ribosomal RNA genes, that was needed in order to read the sequence. So when you grow an organism in the laboratory, it's still hard to sequence its ribosomal RNA genes without this reaction. And that is what people use then to integrate. When you grow something, you go to an environment, you grow as many of the things as you can from that environment, and then you characterize them experimentally in the lab or by this typing. This typing, of course, is not all you want to do. So what you want to do frequently is experimentally study them and do some biology of the culture in the laboratory. And that's what I'm going to walk you through for the next few minutes, some ways that people do this with extremophiles. So let's say you're interested in organisms that grow in very high salt conditions, like I am. You go to a particular environment, you try and culture a particular type, and you take a clone of that organism. That is one individual cell that you've now grown up. And we can do things like see what its favorite salt concentration is. What is the preferred salt concentration for that organism in terms of its growth properties? So you can take multiple test tubes and put different amounts of salt in each of those test tubes and keep everything else identical. So 0.5 molar, 1 molar, 1.5 molar, 2 molar, etc. For organisms, if we're interested in their temperature growth, we can do the same thing with different temperatures in those test tubes. So very controlled conditions, comparing only one variable, say salt. And then we measure how they grow in those conditions. So here's more detail of how you do this. You go to this high salt environment, you grow organisms in the lab from that environment, and you grow up what's called a starter culture. A big test tube filled with the clone of that organism. You set up some flasks with different, with media that they can grow in. You make the media basically identical in those different flasks. You add a little bit of your starter culture to each of those flasks, and the way you study the variation in what this organism likes to do is in each of these different flasks, you can put slightly different salt concentrations. So let's say flask 1, 2, 3, and 4, we do 1 molar, 2 molar, 3 molar, and 4 molar salt concentration. And everything else is the same. We grow them at the same temperature, everything else is the same. And now we watch as growth occurs in these tubes, and we quantify how much growth is occurring. And we can do that frequently by looking at the density of stuff in that culture. The more cells there are, the harder it is to see through it, and we can measure that with a spectrophotometer or other equipment in the lab. So we monitor growth over time. Say after 1 hour it looks like that. After 2 hours it looks like that. After 3 hours it looks like that. We quantify that growth in each of these conditions, and we plot it. So here's the growth rate of that organism in 3 molar salt. Here's the growth of that organism in 2 molar salt. Here's the growth of the organism in 1 molar salt. And now we can plot this slightly differently. We can calculate a rate of growth. That is the amount of growth over time. And we can plot that versus salt concentration, and for that plot, this is what you get. So this particular organism has its fastest growth rate in 3 molar salt of the conditions that we used. And that's how we figure out, in essence, what its ideal growth conditions are in the laboratory. And that's been done with an incredible diversity of organisms relative to their salt preferences. Organisms that can sort of deal with a little bit of salt but not particularly well are called halotolerant. Organisms that seem to really like salt are generally called halophiles. And in organisms that can only grow in extremely high salt concentrations are called extreme halophiles. And in essence, all of the extreme halophiles on the planet are archaea. They're from one branch of the archaea, actually. So one monophyletic lineage of archaea, they are all extreme halophiles in that group. There's some really interesting stuff you can now do by comparing the biology of these organisms, again, usually in the laboratory. So you can try and figure out how they're able to grow in very high salt concentrations. When you put a normal organism like E. coli into high salt concentrations, it is very unhappy because of the osmotic pressure. If you have more salt on the outside, water will want to leave the cell, and you will get the cell desiccating in those conditions, losing its water. Organisms compensate for this, the ones that can grow in high salt concentrations, by filling up the inside of their cells with various what are called solutes, various compounds that counteract that osmotic difference between the inside and the outside. And if you look at the different halophiles, people have found all sorts of different things they fill up their cell with in order to deal with this. Some fill them up with proteins, some fill them up with carbohydrates, some fill them up with salts. I'll come back to that in a minute. Many of them also use a lot of their energy to run pumps in their membranes to try and deal with this osmotic pressure. And many of them are also very resistant to desiccation because they sometimes can't deal with the osmotic pressure. One of the really interesting things is those extremely halophilic archaea are the only ones that fill up the inside of their cell with salt instead of with proteins or carbohydrates. This is very important for biotechnology because a lot of people would like to run various reactions, say laundry detergents, when you have a lot of salt around. The enzymes from these extremely halophilic archaea work really well even when there's lots of salt, because they're used to having salt inside of their cell. So this has been taken advantage of in the biotech industry extensively. As I said, this is one monophyletic lineage of archaea that has figured out this particular adaptation. And all members of this group are extreme halophiles. So we can do the same general approach with growth at different temperatures. We can go to hot or cold environments, take organisms from those environments, do this starter culture. Yeah?
Sorry, if they're archaea, why are they called halobacteria?
They were called halobacteria before archaea were discovered. So some of them have retained that name, unfortunately, because of the old classification of them. You set up flasks again. Now you grow those different flasks at different temperatures. But it's the same general approach. You now measure growth over time at different temperatures. You plot that growth. You calculate a growth rate. And now you have an optimal growth rate of an organism versus temperature. So you can call this the optimal growth temperature of an organism. People have done the same thing that they did with salt and compared different organisms to each other. Cyprophiles are organisms that grow at low temperatures. Mesophiles are the ones like us that grow at the middle temperatures. Once you get above 60 degrees Celsius or so for your preferred growth temperature, that's called a thermophile. When you get above about 80 degrees Celsius, those are called hyperthermophiles. And if you get above about 90 degrees Celsius, the only organisms that are able to do that are archaea. The only organisms that are able to be hyperthermophiles are bacteria and archaea. There are no eukaryotes that grow optimally at temperatures above about 60 degrees Celsius. Yeah?
Do the archaea accomplish that by filling their cells with ions as well?
No, so growth at temperature leads to different stresses than growth at high salt. So what organisms do to grow at very high temperatures is very different. They have to change the amino acid sequence of all their proteins. They have to change their membrane structures. They have to change how their DNA is packaged. There's a huge number of adaptations that organisms do to grow at higher temperatures.
Because wouldn't the water inside of them be boiling by that point?
Yeah, so there are some organisms that grow optimally at 106 degrees Celsius, and you might think that the water would be boiling, because the boiling point of water is 100 degrees Celsius on the surface of the planet. These organisms are from the bottom of the ocean, where the pressure prevents water from boiling. So it turns out the upper limit of growth of organisms is basically where water is a liquid, not temperature. So when people compare growth temperatures of different organisms, as I was mentioning, the archaea grow at the highest temperatures. The bacteria are able to grow up to about 90 or 95 degrees Celsius, but not at 106. And eukaryotes don't grow at the highest temperatures. There's some that grow at up to about 60 degrees. Yeah?
Is the reason why eukaryotes don't grow in those high temperature environments are very rare in terms of like how much area they cover?
No, they're actually, in the bottom of the ocean, they're all over the place. In certain places, I don't think that that's the reason. We can talk in the next lecture a little bit about what some of the reasons might be. What we're going to do in the next lecture is answer your question and talk about it in relative to the human microbiome, which is how we use DNA to study microbes in the environment. I got to answer this, yeah.
So basically, Lowe's' biggest discovery was that he essentially founded the archaea domain.
He did two things.
Via sequencing the ribosomal RNA. Would that be like sufficient answer for like his?
Yeah. So the two
achievements?
things that he did were he discovered the archaea,
Yeah.
and he showed that you could actually classify all organisms on the planet by building trees.
But most importantly, it was microbes, because prior to his discovery, microbes were poorly.
Yes, absolutely.
Okay. Awesome. Thank you.
Yeah.
So with the